This program is a presentation of UCTV for educational and non-commercial use only.
So welcome. Thanks for coming tonight. My name is Jason Pomerantz. I'm a plastic surgeon assistant professor here at UCSF, and I'm going to talk for the first part, and then my colleague Dr. Mankani will continue. This talk is entitled Stem Cells Growing New Parts, and we're going to give you a little bit of background about stem cells, which you may know already, but then we'll talk a little bit about what we as plastic surgeons are interested in doing with stem cells, both clinically and in the laboratory with a view towards future applications. And I think both of our talks will really focus on the therapeutic potential of stem cells. There are other very important uses for stem cells. In vitro modeling, meaning for example, quote, disease in a dish, where stem cells are used in the laboratory to model disease based on how those cells behave compared to similar cells from a normal, healthy patient. In addition, scientists use stem cells to acquire basic knowledge of human development. But really, we're going to be focusing on this application here. So just by way of introduction, stem cells are, in general, thought to be unspecialized cells. And they have the ability to undergo what's called symmetric division, meaning they can divide and make two daughter cells that are essentially identical to them. And those daughter cells are stem cells, which can keep renewing the stem cell population, or they can differentiate and become a different type of cell, which can be a progenitor, which expands and then makes a functional cell type that takes part in a tissue, such as muscle, blood, for example. And once cells get to this stage, they no longer have the ability to self-renew, so they're no longer stem cells. And what has really captivated us in the medical field about stem cells is that the ability to have a cell that can self-renew and not go and differentiate really theoretically provides a unlimited source of cells. And so it's essentially a lot of raw material for use clinically or in the laboratory. This is a schematic of stem cells, and stem cells in the original sense existed in the embryo, the so-called embryonic stem cells. And these cells can give rise to any cell type in the human body. And as the embryo becomes a fetus and later a child or adult, the ability of cells within that body to become many different cell types diminishes. And we'll talk about it later, but even the adult has stem cells. They're just limited in their potential in that they can only become certain cell types, not everything. And that has practical implications. More recently, there have been technological feats which have allowed us to create in the laboratory stem cells. So instead of taking them from an embryo, people have used artificial methods to take a cell from an adult body and either transfect it, meaning place or activate genes that were not otherwise active in the cell, or take the nucleus of that cell and put it into an egg, and then rederive cells that are like embryonic stem cells. These are some of the main players in the history of stem cell biology, people who really started the field and have gotten us to where we are today. And I'll just mention them briefly. This is Gail Martin, who was at UCSF and really, you can say, at least coined the term stem cells and discovered mouse embryonic stem cells. Stem cells in the mouse embryo that could give rise to all the different cell types. And the whole field really expanded from there. This work was in the early 1980s. Human stem cells were discovered later. And then the method that I talked to you about briefly a minute ago, where adult cells can be taken from the body and certain genes activated in them to then derive embryonic-like stem cells, was done by Shinya Yamanaka, who also is an affiliate here at UCSF. Other techniques that really led to stem cell work were first introduced in the 50s and 60s by scientists like John Gurdon, who was the first person to really do cloning. He took nuclei from frog cells and put them in frog eggs that had the other nucleus taken out, and was able to derive cloned frogs. And that work was in the 60s, and it wasn't until decades later that similar experiments were done in mammals. This is Dolly the sheep that you've all heard about, the first cloned mammal. And it was really a technological problem that took from the 60s to the 90s to really solve. And once it happened, it opened everyone's eyes to the possibility of doing many different things with stem cells. And then, of course, there's a lot of ethics and politics surrounding stem cells. And you all know a lot of the debate, and a lot of famous people who have personal involvement with stem cells have really driven the field. So what are the clinical applications of stem cells? Either human embryonic stem cells or adult stem cells that I'll talk about. Well, cell therapy, that means taking a cell that was a stem cell and putting it into a patient and asking it to become a certain cell type and provide a function. Drug screening, toxicity screening, disease mechanism, these are all the disease in a dish applications that I mentioned briefly. These are laboratory applications where, by virtue of the ability to take cells from a patient, make a stem cell from them, expand those cells, there's, in a sense, an unlimited supply of cells from a patient to then try to understand how their disease works, try drugs on it before trying it in a patient. Again, we're going to be talking about the more directly clinical applications. One of the main problems or hurdles with stem cells is that when you start with an embryonic stem cell, an undifferentiated cell, you have to ask it to jump through all the hoops to become the tissue that you want, either a vascular cell, blood cell, a muscle cell, a heart cell. And you have to figure out how to make it do that. And in a sense, stem cells are like a whole group of untamed horses. You have to do a lot of work to get them to become what you want, and there's always going to be a few in the group that are just not going to follow. And that becomes a problem when you're talking about putting cells into patients, because even a couple of bad apples, so to speak, could cause problems. I mentioned these two people already, and what their work has done is help us get around one of the problems, that is the fact that embryonic stem cells, which are derived from an embryo, if put into another patient, would not be immunologically compatible. And so that patient would have to have immune suppression through their life to keep those cells that came from another individual. These techniques make it possible to make stem cells from each individual patient's own body. A skin cell, for example, taken from a patient, can then be placed, the nucleus of which can then be placed into an egg and reprogrammed to become a stem cell, which can then be put back into the patient, and it will be a self-derived cell which won't cause an immune response. And that technique is called somatic cell nuclear transfer. A cell is taken from the patient, any cell type. The nucleus is taken out and put into an egg that has to be gotten from a surplus of eggs. And then the cytoplasm, all the material that exists in that egg, will reprogram that nucleus, which was once doing a different function, to become stem cells, which can then give rise to an individual. And this has been performed in mice and sheep, and as far as we know, not yet in humans, but theoretically there's no reason why it couldn't be. But there are a lot of ethical issues, of course, that surround doing this in a human patient. The other way to get around the immune problem and the ethical problem of using oocytes is to not use either, to, again, take cells from your own body, which are differentiated, and then activate or inactivate certain genes, which will then cause those cells to be reprogrammed and become stem-like cells. And that's the so-called induced pluripotent stem cell. Induced meaning you made it happen by changing the expression of genes. Pluripotent meaning it can become many different cell types now, stem cell. And Dr. Yamanaka did that work first, and it was published in 2006, and many others have advanced and replicated it. And it has changed the field as well. So what are some problems with all of these different types of stem cells? Well, I already mentioned one, which is you have to get them to differentiate, meaning become the cell type that you're interested in. And you have to figure out all the signals, the factors, the molecules that stem cell needs to be exposed to become your cell of choice. They have a propensity to form tumors, and it only takes a couple of bad cells to form a tumor, and that causes a problem for the patient. Product consistency. If you're going to make muscle cells, you need to be able to get muscle cells every time, and not other cell types. You have to know what you're introducing into a patient. Immune response we discussed. And then one of the really key things is integration, meaning, okay, I have a bucket of cells, and let's say I can make them become whatever I want. That's a far cry from becoming a body part. A body part has a skeleton, it has a scaffold, it has a shape, it interacts with other body parts, and cells are one component of that. It's almost like saying, okay, we've invented steel, now go build a car. You have to know a lot more, and you have to be able to do a lot more than just have the cell of choice. And I think I'll talk about one approach that we're using in my lab to look at these problems, and I think Dr. Mankani will talk about a different approach, both of which take into account the really difficult problem of integration. So we talked about embryonic stem cells and ways to make stem cells to induce them, but I want to make the point that we all have stem cells in our body. Everyone in here has stem cells. And how do we know that? Well, this is really a timeline starting with year one and a person to about the age of 70. And you don't have to read the words, but all these circles represent different tissues and how frequently they turn over. So for example, every bone in your body totally replaces itself every 10 years. Your skeletal muscles replace themselves. Your skin replaces itself much more frequently than that. Here's the liver. Every 3 years, completely replaces itself. So that has to happen by stem cells existing in each of those organs that continually maintain the turnover and homeostasis of that organ. They're different from embryonic stem cells because they seem to have a more limited function, meaning a liver stem cell probably only makes liver. It doesn't make everything. But there's a huge effort by many groups to harness the potential of these cells because, for example, why derive an embryonic stem cell and try to make it go through all these hoops when you're trying to make liver, why not use a liver stem cell? So those are some of the issues and thought processes. Adult stem cells are important in our daily maintenance of tissues. They become very important in pathological situations, in trauma. So if you have an injury that needs to be repaired, that tissue needs to be rapidly replaced and turned over, and stem cells are responsible for that. And that's also where, in humans, some of our ability stops. So we have excellent ways to heal wounds, to close wounds, but if I lose my bicep from a surgical procedure or I get shot, it doesn't grow back. I'll get scar and my wound will heal, but I will not get regeneration. So the stem cells that we have there can do some amount of homeostasis and maintenance for me, but they cannot regenerate a structure, which brings us back to the goals of what we're doing as plastic surgeons. Are stem cells that we have in our body useful therapeutically at all? Absolutely. So this is the quintessential stem cell, a hematopoietic stem cell. This is a blood stem cell, and it was the first one that was really discovered in the adult animal. And hematopoietic stem cells maintain all the different types of blood cells in our body every day of our life. They turn over our red blood cells, white blood cells, and you can take one hematopoietic stem cell and inject it into a mouse and reconstitute that entire mouse's blood system just from that one cell. This is the first therapy that was done with stem cells, bone marrow transplantation in humans, and that started a long time ago. That's not a new procedure. It's not a new concept. In the 60s, it was started, and in concept, and it really took off. These are the number of transplants performed per year. And this is a major medical therapy, and it's stem cell therapy. We might say that compared to a solid organ, perhaps replacement of the blood doesn't have all of the same challenges. It's a fluid organ, which really requires circulation of its cells. It doesn't require the same type of three-dimensional, rigid structure that solid organs do. In plastic surgery, we are using stem cells for a number of applications, and we're just going to mention a few. Here's an example of one thing we do. So this is a patient with a soft tissue deformity. You could see that the asymmetry in the face here. This is the normal side, and here he's had atrophy of all the soft tissue on that side of the face. We transplanted fat cells from this patient, from his abdomen. Here they are in these syringes here, with tiny little injections over time. And those cells survive in the place where we put them and replace the soft tissue defect there, and they recreate the symmetry. Here we're about 20 years behind the bone marrow and blood transplantation folks who have since identified the single cell in the blood system that is responsible for allowing this to survive over time. Here we're just transplanting large amounts of fatty tissue, which we believe contain adult differentiated fat cells, in addition to probably some fat stem cells, which over time are responsible for the long-term maintenance of this correction. Many of our efforts are currently underway to identify the stem cells that exist in adult adipose tissue to more predictably and more easily correct defects such as this. sorry for the images, but these are muscles. This is the rectus abdominis muscle. This is the latissimus muscle in a patient's back, and this is just to give you an idea of what the challenges are in using a cell therapy in a solid organ. So, one challenge is even to get our stem cells into a tissue like this to change its function. But a whole separate issue is, how do you make this? So the stem cells are one part of it, but again, there's a lot more here than just cells. So growing new parts is a very complicated and long-term goal of plastic surgery. And Dr. Mankani and I are interested in addressing problems for which we don't really have great surgical solutions yet. We can do a lot with surgery. We can move any of these muscles around. If my bicep is gone, I can take a muscle from the back and put it in the bicep spot and hook up the nerves and the vessels and get the elbow to go like this. We can do a lot. We can't replace an arm. We can't replace a part of the face that's missing. And it would be nice to be able to remake a bicep and not have to take a muscle from the person's back. So again, the humans don't regenerate very well when we're adults, except for certain organs like the blood and the surface of our skin, which replenishes a lot. But if you have an injury, it doesn't grow back. This is a patient who had surgery on the skull for a deformity, and that left a hole here, and this hole doesn't fill in. If the patient were an infant or an embryo, the hole would fill in no problem. But once we get to be adults, we can't really regenerate. This is Jerry Garcia, the famous guitarist. He has an amputated finger, doesn't grow back. So although we have a large capacity to regenerate some organs like the blood, we don't regenerate many others. So who does? Well, this is a newt, and newts are vertebrates like us. They have backbones, and they have all the same types of tissues. They have nerves, bones, muscles, skin, and they regenerate remarkably well. This is an example of newt regeneration. If you take an adult newt and you amputate the arm, either here at the elbow or here farther up near the shoulder, over time, it grows back. And it grows back with a perfectly functional limb and hand. And there isn't really much of a difference in complexity here between the newt limb and our limb. Ours is bigger, of course, but this has bones. It has digits with little bones in each finger. There are tendons, nerves, muscles, and all the same structures that we have. So what's the difference? Why can these organisms regenerate, and why can't we? And that's one of the focuses of my laboratory now. And I'm attracted to looking at these different organisms and understanding why they regenerate, because all of that work that we would need to do to take a cell, which we have in a dish, and convince it to become a certain cell type, and then take that cell type and put all the other components together to make a tissue, is extremely difficult and promising, but extremely difficult. Here it happens naturally. So if we can understand why these organisms do it and why we don't, perhaps we can mimic the scenario in our bodies in certain situations to affect regeneration. So in the newt, also in many types of fish and other types of amphibians, regeneration happens by a different mechanism than what I described as stem cells. So stem cells are cells that sit in certain parts of our body waiting for the need or an injury, and then they proliferate and make the new tissue type. In these animals, when an injury happens, these are muscle cells here, and you can see the striations. That's the mechanism for the contraction of the muscle, and these are nuclei, which contain the genetic material. When an injury happens in newt muscle, those nuclei, which were inside the muscle cells, not stem cells, change their function. They say, hey, okay, there is an injury, now I have to do something. So they bud off of the muscle cells as cells and start to divide and proliferate. So essentially all the cells at this site of injury change their function after an injury and become stem-like cells. They all proliferate, and they make what's called a blastema, which is a mass of cells that are undifferentiated. This is cartilage and bone here, and after an injury, again, all those cells at that interface change their function, proliferate, and make this blastema, which then somehow knows to proliferate very rapidly and in a controlled way so that it can then, each cell will then differentiate again into the cell type that it needs to replace the missing part. Yeah?
I have a question. Yeah. So are these stem-like cells pluripotent? Because
No, certain really elegant experiments were done in the last couple of years with lineage tracing by Eli Tanaka, who traced the cartilage cells, the nerve cells, the muscle cells, and they're not pluripotent. They simply gain proliferative potency. So they go back into the cell cycle and grow, but cartilage doesn't become nerve, nerve doesn't become muscle.
so it remains in the same cell line.
Same lineage, yeah. So I began asking these questions in mammals, and I did this work with Helen Blau at Stanford. Muscle cells in mammals are a good system for studying differentiation because they're well characterized. Myoblasts are muscle cells that proliferate that are not yet differentiated. They're not quite a stem cell because they can't be quiescent. They can't sit without dividing. They grow rapidly, and when they're put under the right conditions, they exit the cell cycle. The cell cycle is how a cell grows and divides. They exit the cell cycle, meaning they become post-mitotic. Mitosis is division. They no longer divide. They become post-mitotic. They differentiate into what's called a myocyte, and they fuse with each other to become what's called a myotube, which are those long structures that you saw on the Newt slide, which are the differentiated muscle cells, which have the function of muscle. They contract and they make parts of the body move. This is not reversible in mammals, unlike what I just showed you in newts, unless sometimes in the case of cancer. But you don't go backwards. So why can't our cells go backwards like the way the newts do? Well, we looked at what controls growth and differentiation in newt muscle cells and compared that with mammalian muscle cells. So I'm just going to go through this quickly, and I don't want to bog everyone down with details, but RB is a protein. It stands for retinoblastoma, which is a tumor suppressor. And it's a tumor suppressor, and it's also a protein that controls that cell cycle exit, even under normal circumstances. So we all have RB in every one of our cells, and every time one of our cells decides to stop dividing, it's because RB is controlling that. When phosphorylation happens to RB, it lets cells proliferate. And this is exactly how newt muscle cells go back into the cell cycle and start growing again. Their retinoblastoma protein gets phosphorylated, and boom, they proliferate. So we tried this in mammalian muscle cells, and this is the slide came out dark, and there's a lot of things here, but what we did was we temporarily inactivated retinoblastoma in muscle cells from a mouse in culture. These red long cells are the muscle cells from a mouse, and we asked whether they started to cycle again. And if they did, this would all be lighting up green. They didn't. So that's a fundamental difference between our cells and those of the newt. You inactivate retinoblastoma in the newt muscle, they grow. You inactivate retinoblastoma in mammalian cells, nothing happens. So why is that? Well, if you look back at how growth control is affected in these animals, these two pathways are evolutionarily ancient. Retinoblastoma, which I just told you about, and P53. P53 was named Molecule of the Year some years back. It's the other most important tumor suppressor, and it exists in all organisms. Everything back to yeast has P53 and RB. These are the two main growth control pathways in cells. And lower vertebrates have these, as do we. But we have more complexity. So we have retinoblastoma and we have P53, but we also have an extra evolved tumor suppressor gene called ARF, which those organisms don't have. And what ARF does is it helps RB and P53 talk to each other. If RB gets inactivated when it shouldn't, like in a cell of yours that gets mutated because you're in the sun, ARF gets activated and signals to P53 to shut that cell down. And this is a feedback mechanism which we found does not exist in newts and other organisms, other vertebrates that regenerate. So a hypothesis that we brought up was, did we develop the ability to suppress tumors at the expense of being able to regenerate? So are we being a lot more cautious about letting our cells grow? And this is teleologically speaking, of course, to protect us from getting cancer. And so, well, what happens? Well, we looked, and when we inactivated RB, so here, this is showing expression of RB in cells. And here we used a genetic trick to inactivate RB. And when we do, ARF gets induced. ARF gets expressed, which is what we expect from that diagram that I just showed you. So what happens when we knock down then this fail-safe mechanism? We knock out RB, we knock down ARF as well. Well, all of our nuclei then will proliferate and go back into the cell cycle. So, and that's shown here by the green cells in these muscle cells. Here is just RB, here is RB and ARF. So this was done to mimic the scenario in a newt. We asked, if we just manipulate these genes and take out ARF, because we know ARF doesn't have this gene, can we get proliferation to occur? And sure enough, it did. This is not projecting at all, so I'll skip over it, but this is showing that the phenotype, the structure, and the gene expression that occurs in the cells also changes in addition to the ability to proliferate. This is tubulin, the skeleton of the cell, before and after loss of both RB and ARF. The cells completely change their shape. And here we used a very fancy technological trick that allows us to identify single cells. So these are single muscle cells that are differentiated. We removed RB and ARF. We used a laser beam to circle the cell sitting on its membrane, and a burst of laser beam light called pressure catapulting, which flicked this single cell out of the dish into its own dish so that we could watch that cell over time and see if it proliferated, and if it proliferated. And to do that very rigorously without wondering whether another cell from a different area crawled in and is confusing the picture. And if you look down here, if we just use controls, meaning we don't actually take out RB and ARF, none of these proliferated. They just crawl off the little membrane and they sit there. But if we knocked out or down RB and ARF, these cells grow and form colonies. And they reactivate genes of a more premature or immature muscle cell type. When we put them back into live animals, if done under the right conditions, they incorporate into the muscle. All these little green specks here are the muscle cells that were in a dish that are faithfully reincorporating into muscle cells. So we were able to recapitulate in mammalian cells the same behavior that newt cells do when they regenerate. And we were able to go backwards here by removing a gene that is a tumor suppressor in us but doesn't exist in those organisms, and so is a really good candidate we say regeneration suppressor. And again, the whole goal is to see if we can get back to this stage in mammals. This is that blastema from the newt cell with the regenerating limb. So mammalian muscle cells can de-differentiate, go backwards, and proliferate. And the approach mimics a mechanism that already exists in nature. We're really just trying to see if we can induce our tissues to behave like our other vertebrate counterparts can. And it suggests really that in evolution, we lost some regenerative potential at the expense of gaining tumor suppression. And our focus of what we do is to ask whether we can approach regeneration the way these organisms do. So this is a heart. After a heart attack, you lose part of the heart muscle, which dies. It never regenerates. There's no stem cells in the heart. Once you lose a piece of heart, that's it. Those organisms can regrow their heart. You can cut half of the heart out, they'll form a blood clot there, and the heart will regrow. And it does so by the same mechanism that I just showed you. So we're trying to reawaken that mechanism in mammalian cells. And that's a major focus of our work as plastic surgeons. We don't limit what we do by a specific body part. We try to solve structural problems, and we try to use innovative techniques. So with that, I'll turn it over to Dr. Moncani, who will give you another perspective on what we're doing. And then we'll take more questions at the end. Thanks.
Well, I'm going to take a slightly different tack this evening and actually come at this from the other direction. Dr. Pomerantz gave a very nice talk about stem cells and the basic biology that regulates them. I'm going to actually show how this biology might be useful from a structural standpoint in reconstruction, in tissue engineering specifically. So tissue engineering is actually the incorporation of three different areas together to make tissue. The first is the incorporation of cells, and that can be stem cells, or it can be cells that are already differentiated and committed along a certain lineage. And those cells need to reside on some sort of template or scaffold which helps hold them physically close to each other, which may actually serve as a structure that regulates how they function, but also will allow them to attract the vasculature and to perform their function. And the third component is growth factors. The growth factors are going to help regulate the cell function as well. And those growth factors may come from the recipient animal or person that the cells are within, or they may be exogenous growth factors that are administered at the time the tissues are placed, the cells are placed. What I'm going to do this evening, just for shortage of time, is focus on the issue of cells and a little bit on the issue of the scaffolds and show how they interact together to help us solve problems. Now, the cells that we're talking about are cells that are very similar to what Dr. Pomerantz was mentioning, either cells that are already committed, which are differentiated cells, or cells that are not yet committed, whether they're adult-derived stem cells. They can be embryonic stem cells, or they can be the induced pluripotent stem cells. The latter three categories are cells, again, that are not yet committed to a particular function. And as Dr. Pomerantz mentioned, the definition of stem cells are those cells that can renew themselves through successive cell division so that you still maintain a pool of uncommitted cells. And these cells also have the ability to be induced to take on certain organ or tissue functions. What we're going to focus on this evening is adult-derived stem cells, which is the area of my research. So from my standpoint, the advantage of using adult stem cells for different tissue engineering modalities are just listed here. Major advantages are that in the case of cells being taken from somebody and then developed in the laboratory and then placed back in that patient, these are then cells that have very little risk of transmitting diseases or having immunologic problems associated with them. These cells, for the most part, can be tailored or trained or directed towards certain phenotypes. The cells can be delivered as either discrete constructs, such as trying to make a new organ or a new tissue type, or they can be distributed systemically throughout the animal or the individual to take on a certain function. And then lastly, the cells can be used as a vehicle for gene therapy. For instance, a patient that is not producing insulin, is diabetic, theoretically you could tailor cells to express insulin, and those cells could then be placed into the patient, and then when the patient gets their drink of Coca-Cola, they make sufficient insulin to respond to that. That would be an instance where gene therapy might be useful, using cells to administer that gene. The disadvantage with a cell-based therapy is that it typically requires the expansion of the cells in tissue culture. And what that means is we may identify a few stem cells in a patient, take the stem cells out, and then we need to expand that number, increase the number to a point where they are useful to the patient. And that can take time to do in the laboratory, maybe on the order of weeks. So if somebody comes in with an acute catastrophe, they just had a heart attack, they have dead heart muscle, as Dr. Pomerantz was mentioning, you may not actually have the time to deliver their own expanded cells back into them. So the disadvantage here is time for autologous cells. Likewise, these cells are typically not really appropriate for an off-the-shelf product. That is to say, something that can be taken off the shelf in the hospital, opened up, and just given on the spur of the moment to a patient. You don't really have that capability with cells that are expanded in tissue culture. This is a disadvantage as well for sort of broad applicability. Typically, these cells require some sort of hospital environment to be used, so it's difficult to get them involved in your doctor's office or your dentist's office. Despite the fact that they usually go from one patient back into the same patient, there's still that risk that they will serve as a method for infecting the patient. They may serve as a vector. If the cells become infected during the preparation process, during the development of the product in the laboratory, they can then transmit that infection back to the patient they're trying to treat. And then lastly, even though these cells are derived from adults, and so our stem cells would still have minimal chance of becoming malignant, there is that possibility. And so anytime cells are taken out of the patient, expanded, and then put back in, there's concern that they may become tumors. Now, the area that I'm interested in, which is taking cells from the bone marrow and expanding them in tissue culture and utilizing those properties for tissue engineering, actually started with the work of this man, Alexander Friedenstein, was a Moscow scientist who in the 1960s and 70s was the first to understand the potential of cells derived from the bone marrow. And what Friedenstein did was take preparations of bone marrow out of donors, whether they were animal or human. He would plate them into tissue culture. He would put them into tissue culture flasks and look at them over time. And he found that there was a population of cells in that bone marrow which would settle down and adhere to the plastic or the glass surface of the tissue culture container. And that over time, these cells would divide. They would proliferate. And they would divide either as individual colonies, which would be clones. So a single cell replicating itself over and over, all the same genetic material, but a population of that. Or they would become populations of clones. And he would then combine these cells together with some sort of matrix and put them back into an animal recipient. And when he did that, he found that the cells actually took on different characteristics. Some of them went ahead and became bone, some became muscle, some became cartilage. And he spent quite a bit of time trying to understand how those pathways were regulated. Why is it that some cells went down one pathway, some went down another? But again, he was able to, again, characterize something and explain something that before hadn't really been recognized. So the characteristics that he was able to describe were that, again, these cells were able to adhere to a tissue culture flasks, that they had characteristics of what we call fibroblasts, which are cells that flatten and have a certain appearance in the tissue culture. And again, that these cells were clonogenic. When he put these cells, when he took them from the tissue culture flask and put them into the animal, which is what it means to go from in vitro to in vivo, he found that they were not the red cells or the white cells that we see in the blood. They were not that portion of the bone marrow, but they were a different population. And that this population was one that would divide again into become bone, would become cartilage, could become muscle. So we've had a chance to extend that work, first by using the initial models that he described, but going further to try to come up with a system for producing bone in a very regulated, predictable fashion, with the idea of potentially treating patients who have bone disorders or bone deficiencies. And what that involves is taking bone or bone marrow as an aspirate, taking it as a suction aspirate from the bone marrow, putting that in tissue culture, getting the cells together, combining them with an appropriate sort of scaffold, and then putting them into the recipient mouse. And what we find is that the appearance of the bone, after a certain point, in this case 9 weeks for this picture, is very similar to what we see of the normal bone that comes from that animal in the first place. So this bone has cancellous, it has trabeculae, which are the small components of bone, and they have a histologic picture which is very similar to that what we see in normal bone. They form a bone marrow. The bone marrow has red and white elements, and it has fat. And when we look to see whether or not that bone that's developed has come from the donor, the human that donated the bone marrow cells, or whether it's come from the recipient animal that the cells are placed into, we're able to confirm through tests on the cells that they actually come from the donor animal, from the donor human. In this case, it's a marker. It's a gene sequence in human cells which is not present in mice, and we can detect that gene sequence. And when we do, it stains the cell black, and that's what we're seeing in these sections that are coming out of a mouse. So how do we go ahead and use this? Well, there's a large population of people in the community that have fairly substantial deficits of bone in their skulls. And this young man is an example. He's a patient of mine who was mugged and was actually beaten about the head. And the neurosurgeons, in trying to save him, realized that he had a lot of brain swelling. And the current technique or current treatment for that swelling is to go ahead and remove a portion of the skull, as was done here, and bank it or put it away in a freezer, and then bring him back after he's recovered from that injury and put that bone back in its place in the skull. Unfortunately, what happens is that there's a great deal of unpredictability in terms of the viability of that bone, and oftentimes it'll resorb or get absorbed by the body, so that the patient will be left with large voids. And in his case, he had these large contour deficits, where if he turned his head, you'd be able to see that he had a large sort of indentation. So, and this is actually a fairly common phenomenon for these patients. And in addition, this is a problem that occurs not only with patients in our communities, but it's a consequence of the two military actions that our troops are involved in overseas. So when these patients end up with large deficits of bone, the traditional treatment is to go ahead and take bone from elsewhere in their body and lay it in. That bone either comes from their ribs or it comes from their hips. But if the bone deficit is substantial, like in some of these patients, that can really debilitate them in another area. So if we could develop a way to fill those bone defects by using their cells in a sort of minimally invasive way, that would be an advantage to these patients, and that's what we've been trying to do. The other approach that we've taken with this is not just to focus on the calvarium, on the skull, but also to start thinking about ways of creating multi-tissue constructs. So in the previous situation, I was showing you really a technique that we're trying to develop for just replacing bone by itself. But but that actually has very limited applicability. I mean, there are those patients, but we have actually other patients that come in with more complicated deficits in the mandible, for instance, where they don't have all the bone around their jaw. And reconstructing a mandible turns out to be a much more complex problem, where we not only have the bone loss, but this bone itself is weight-bearing. So it's got a great deal of intrinsic strength to it. And reproducing that is a challenge independent of the challenge I described in the calvarium. And this mandible also supports teeth. And so how do you reconstruct a tooth? And the mandible has incorporated in it a joint, the TMJ, which articulates with the skull, which means it has bone, but there's cartilage sitting on top of it. And then in addition, it has a blood vessel and a nerve that run inside, which perfuse it and help give sensation to the tissues and to the teeth. So how do you go ahead and incorporate that? So we can produce one tissue, bone, we can produce cartilage, we can start producing vessels, but to produce all of them in concert with each other is a challenge that we haven't yet been able to surmount. And there are a substantial number of patients with that sort of problem. This is a cartoon from the 1900s. You may recognize it, Andy Gump, who had no mandible. And we have patients that have an Andy Gump deformity. They've had their mandible removed due to cancer, due to a cancer operation. And we have ways to go ahead and reconstruct that. And we can take a piece of their leg bone, their fibula, which we can then carve into the appropriate shape and attach with microvascular surgery back into the mandible, into the space where the mandible was. But this is very debilitating surgery. If there's a way that we could do this without having to put the patients through it, it would be a big advantage. So if you think about engineering tissues, engineering the mandible, we've got multiple tissue types. We have bone, tooth, the TMJ, blood supply, nerve, covering, which is skin and mucosa. We've got challenges in terms of where to create it. Do we do it in the patient? Do we do it in the laboratory and transfer to the patient? If we're going to do it in the patient, do we do it in the face, or do we actually do it in another site and then transfer from the other site into the jaw? I mean, these are all real technical issues as surgeons that we wrestle with. How do we deliver it? If we could put it in through an injection rather than a large operation, that would be much better for the patient. Faster healing times, less pain. So that is also a technical issue. How do we get it to sort of fit in with what's already there? How do we get it to mate with the pre-existing bone? And then how do we put it all together? How do you actually integrate all these little pieces so that we end up with something whole? Well, we don't have clearly time in one evening to talk about all these things, but I'm going to talk a little bit about a few features. Bone in terms of cells and the scaffolds, talk about a little bit of the vascular supply, or in other words, the blood vessels, and the delivery, and just give you a little bit of insight into how these problems have been approached, not just by me, but also by others that are in this field. So talking about cells for just a minute. As I was showing you before, in the mouse, using Friedenstein's techniques, we've been able to form bone from humans in the mouse. So one step towards demonstrating that this is feasible in patients is to show that the bone that can be formed is actually substantial in size, comparable in size and volume to that which we might expect a patient to have a need for. And so here we have a dog experiment where dogs got 2 sets of transplants. One set had cells in addition to a mineral scaffold to hold them in place. And the other side of the skull got just the scaffold without any cells. And we looked to see how those did over an 18-month time period and found that the cells with the scaffold produced substantial bone, and the bone was actually very normal in appearance. And the side that got no cells had some residual scaffold and a lot of fibrous tissue, but really no bone formation. So it's suggesting that over the long term, we can create bone and that the bone actually stays stable. And then in addition, we did these CAT scan studies on the dogs to try to determine whether or not we could use just CAT scans to identify the presence of the new bone. Because clearly, if we're going to try this in a patient, the patient would prefer to have the assurance that there's new bone without having to be rebiopsied. So we were able to successfully show that the CAT scan technique and the special analysis done with that can help confirm the presence of the new bone. Now, if we're going to reconstruct a mandible, we need to make sure that the bone that forms in the mandible is actually capable of filling that defect or of holding in place over time. And so we did a similar study in the dogs in the mandible to show that the presence of these cells, in addition to the scaffold, led to long-term presence of the bone. And here we have an animal that got a transplant at 17 months. It had persistence of that and all that transplanted formed bone. The side that got just a scaffold, that scaffold essentially disappeared over that time period so that it was no longer visible. So again, long-term studies in a large animal to demonstrate the feasibility of the technique is sort of part of the process that we go through to show that these techniques are feasible. Now, we're not limited to the use of cells out of the bone marrow. There's a large body of study now on using cells out of the fat. And there's no shortage of adipose tissue in our population. Every one of us has fat that can be taken out and put into tissue culture in the same way that the bone marrow is. These cells express many of the same markers, i.e. they have many of the same characteristics of the cells that come out of the bone marrow. So they could also serve as a very good source of bone-forming cells. And in fact, scientists have done studies looking at the ability of these cells to heal bone defects in dogs the same way that I'd shown you for our bone marrow study and have shown success in those techniques. There is a lot of interest in looking at the use of embryonic stem cells to go ahead and form bone. And to date, that has been relatively unsuccessful with some very specific exceptions. And one exception has been that of a group at Michigan, led by one of my colleagues, Paul Krebsbach, who has been able to get these ES cells, human ES cells, in the laboratory to be directed towards bone-forming lineage. And when these cells are placed into a mouse recipient, he's able to show that they can form bone. So this actually does have potential. And I think we'll know better as he does longer-term experiments and confirms that tumors are not developing in association with this bone. So we talked about cells. We're going to talk about scaffolds for just a couple minutes. So as I was mentioning, scaffolds are sort of critical to holding cells in place and helping get that organ formed in a three-dimensional sense. There's a real dynamic, though, between having a scaffold which is dense enough, in the case of bone regeneration, to actually support a load. So if you're going to replace a portion of the mandible or a portion of the tibia, you want a scaffold that's really dense so that if someone's walking or chewing, it's not going to fracture. And yet that density is a real disadvantage because it impedes cell perfusion and cell viability within the interior of that scaffold. Cells are not going to get perfused. So there are a lot of efforts going on right now among material scientists to develop scaffolds which are both strong and yet porous. One of the techniques they use is this technique called robocasting, which basically uses inkjet printer technology or laser jet printer technology, where you fill a tube up with the ceramic that you want to use, the mineral that you want to use to make the scaffold, and that's basically squeezed out as it would be out of a toothpaste tube, but it's done through this inkjet printer, and it can be under computer control used to lay down a scaffold with very specific parameters. So in this case, the computer was able to lay down the rows of this in a very predictable, organized way, so that so you build a three-dimensional scaffold. And that scaffold can be then manipulated further. It needs to go into an oven. It needs to be sintered at 1,000 degrees centigrade. But the conditions under which it's sintered can then determine how well it does in association with cells. So here, for instance, the scaffold was sintered at a little lower temperature for a little shorter period of time, and it maintained its porosity, or the presence of the holes and the spaces within that transplant, within that scaffold. But at a higher temperature and for a longer period of time, it became more dense. And these surface features and this porosity have direct action on the cells in determining whether or not they're going to be bone forming and whether or not they're going to remain viable. In the case of these particular scaffolds that I just showed you, we're using them now in the lab to see whether or not they might actually be good scaffolds for supporting bone formation. And this is a study that we completed last year in which we had cells in for a total of a year in these scaffolds. But at 38 weeks, in many of the scaffolds, they had actually filled in many of the voids. And where they hadn't, there was actually the presence of new bone marrow formation as well. So these scaffolds might end up being very conducive to bone formation and may end up being useful in the ways I've been talking about. This process has been taken by other people one step further, using what's called CAD/CAM design, or computer-aided design, computer-aided manufacturing, where a portion of a patient's skull, in this case jaw, is downloaded from the computer after having patients gone through a CAT scan. The data's in the computer. It can then be downloaded and used through the same technique of the robocasting to make a construct which resembles the portion of the anatomy of the patient. And that can be then mounted on these little wires and put inside a machine called a bioreactor. And in the bioreactor, it can sit for a number of weeks, be continually perfused. And if there are cells that are placed on that, those cells will function to start laying down bone mineral. And here we've got an experiment that was published last year in which these constructs were in the bioreactor for 5 weeks. And you can see that over time, they have mineralized this construct fairly extensively, so that it looks much more like the condyle of that patient's mandible. Now, to be truthful about it, this works very well in the lab. Taking this construct out of the bioreactor and putting it into an animal recipient, we haven't been able to demonstrate that it does as well. Part of the problem is now this density issue. This is a very dense construct. It's full of bone, but the cells inside, which have been used to being perfused by this very special bioreactor, don't have any blood supply. And so they actually have no way of becoming perfused while they're inside the animal. So this is, again, a situation where 2 tissue types need to be integrated now. There has to be a vasculature somehow integrated into this design, and that'll be the next iteration on this. The third thing I'm going to say about scaffolds is that we've been talking about sort of constructs which are whole, and yet as we're putting constructs into patients, there are going to be situations where we want to put in these constructs through an injection rather than a substantial operation. And so there's a lot of effort put into developing gels which are able to accommodate cells, and that cell and gel construct can then be injected. And as of yet, these constructs are not yet available. They're still being developed. But eventually, if they can be available, we could use them to inject right into a hole, for instance, in the bone that's developed. Or they could be used to inject right into a fracture. And you could get potentially faster bone regeneration and faster bone healing in that fracture. The next area I'm going to talk about very briefly is the issue of creating a vasculature. So it's been known that without a vasculature, if you simply have tissue in the body, it'll get perfused and it'll get nourished for a relatively short distance, somewhere on the order of 200 microns, much less than 1 millimeter in thickness. So that tissue engineers have spent considerable time trying to design networks that would reproduce the sort of vascular pattern we see in patients. And these networks then can be engineered so that they'll provide a great deal of surface area to any cells that may be in the interstices, so that they'll again reproduce a typical vascular pattern that we see. I'm going to come back to this in just a second. And one of the ways that they do that is, again, through this inkjet technology, you can take cells that line the blood vessels and literally inject them in a pattern on a membrane. And in that pattern, here are the cells and here are the voids, and those voids will become the actual lumens or the spaces of the blood vessels, and what's been laid down will be the walls of the blood vessels. And through this technique, you could theoretically develop a way for developing a vascular pattern. This has actually been taken a step further, where cells have been laid down, allowed to proliferate in the pattern, and then over time, in this case 3 weeks, begin to appear or show the appearance of an actual blood vessel. So we had taken an approach a few years ago that's a bit different than what I'm describing, but again, another example of a way to deal with the need for vasculature, where we actually went ahead and just used the vessels within an animal and formed our bone around it. We put our construct, which was a scaffold and some cells, around that. We covered it with a sheath that didn't permit any perfusion from the outside, and looked at it to see whether there was actually bone formation. And as we expected, there was substantial bone formation inside, and this can serve as a method for constructing a perfused organ. The downside is that it requires the blood vessels as the animal or the recipient, and it's certainly much more elegant to be able to build those into the construct at the same time that the construct is being developed. So although this was one method, we're not thinking it's quite as elegant as what might be eventually available. The one group that's actually made the most progress is a company called Cytograft in terms of producing blood vessels. And actually, this is an example of not of a stem cell technology, but a differentiated cell technology. And they did something actually fairly clever, which is that the two investigators that started this company realized that there's a layer of protein that's laid down whenever cells are in tissue culture over a period of time. And usually as cell biologists, we consider that to be more of an irritant than anything else, but they had the bright idea of using that as potentially as a source of blood vessels. And what they did was they took that sheet out and rolled it up like you would a cigar, and then got it to be lined with the cells that we would typically line blood vessels with. And over a number of iterations of this, got it to the point where actually it could function as a blood vessel in patients. And the way that they were able to use it and show its effectiveness was as a methodology for patients needing hemodialysis. So patients needing dialysis typically need a blood vessel into which the catheter can be inserted. And that blood vessel gets tired over time, it gets scarred and gets infected, and patients need to go to new sites. So they went ahead and put their construct in as a potential blood vessel for hemodialysis, and were able to show in a study that they did in Europe that a number of these stayed open even past a year. And this is something that they published last year, and I'm sure they'll have further long-term data. But this is a potential application where, again, stem cells could be involved if they wanted to go that way, where actually blood vessels can be designed, produced in the laboratory, put into a patient, and potentially be successfully employed. So the last little part I'm going to talk about in terms of engineering a mandible is, how to deliver it, and this issue of how do you put it in? Can you inject it, or do you need to actually do a substantial operation? And we have been looking at techniques to try to deliver our transplant system through a needle. And this isn't quite a needle. This is what's called a trocar, but it's about 4 or 5 millimeters in diameter. And we designed an experiment in which we subjected mice to either an operation with a large incision, which I'm not showing here, or to very, very small incisions. And then we put the transplants in under the skin at these very remote sites from where the incisions were. And then we compared that to the standard technique where we made the large incision. And we actually found that the bone that was formed through either technique was comparable. We found substantial bone. We found substantial bone marrow, suggesting that there may be actually options, even in trying to deliver bone to patients, of using small access ports. So just to summarize a little bit, what we've been able to achieve, meaning people with our interest, are the creation of individual tissue types, whether it's a blood vessel, whether it's bone, whether it's cartilage, which I haven't talked about, or other tissues. That is very feasible. We're able to incorporate scaffolds which are very specific to an application, whether they be dense or porous. But for a particular application, we come up with a scaffold, at least on a small scale. We're also able to generate dual component constructs. So we can put cells and scaffolds together. We can put genetic material onto scaffolds. We can transfect cells so that they have genetic material. We can do these sort of dual constructs, and that works well. But that's so far the limit of what we're able to accomplish. The real challenges are going to come ahead for constructs which are actually clinically useful. For developing large volume perfuse, which means has a blood vessel and blood perfusion, and functional tissues, this is really our next challenge. And that includes the issue of trying to develop multi-tissue constructs, whether it's bone and cartilage, which is a joint, or bone and nerve and artery, as you saw in the mandible. Those are things that we still need to do. So what's next? So I've actually described now to you constructs which are all sort of solid organ reconstructions. I've shown you bone, trying to replace a piece of bone. And I think being successful at that will validate the ability of these cells to be helpful to patients. But I don't really think it stops there. So, our interests actually go a little bit beyond that. Osteoporosis. This is a disease of bone mineralization. There's too much bone resorption. There's not enough mineralization. The bones are weak. This patient of mine who had osteoporosis and fell and suffered a fracture of the radius, had a substantial operation, plates and screws. Wouldn't it be nice if we could avoid the whole issue of osteoporosis? Well, perhaps it'll be feasible at some point to inject these same bone-forming cells systemically throughout the patient, ideally have them home to bone, lodge in those niches where bone cells reside, and go ahead and make up for the deficiency in the bone formation that's been occurring. So this is one area of research that we're starting to pursue. And then the other is a different application altogether for these cells. This is a patient with rheumatoid arthritis, which is an autoimmune disease. Their own tissues are being attacked by cells within the bloodstream. And there are some very extensive heavy-duty medications that can be used to alleviate this process. They have a lot of side effects. Once a patient's reached this point, it's a very debilitating disease. We have reconstructive procedures that we can do for them. Wouldn't it be nice if we could arrest this process fairly early on? And it turns out that the cells that I've been talking to you about, cells out of the bone marrow, when they're delivered not just back into a patient, but actually delivered from one patient to another, actually are able to modulate some of the problems we're seeing in autoimmune diseases. And so over time, we may be able to develop therapies using these cells for patients with autoimmune problems. And I think if that's successful, that'll actually be a real challenge to overcome. The work that I've talked about in my lab actually represents not just my efforts, but that of my collaborators. And one big advantage of being here at UCSF is that we are blessed with a lot of very interesting people with a lot of skills and ideas. And so the ability to interact with them and to collaborate is a huge reason for being here at UCSF. And so I'm blessed with collaborators both in the School of Dentistry as well as over at the VA, as well as collaborators at Berkeley, and then my old lab back at the NIH, who I still work with. Thank you very much. So I think that Dr. Pomerantz and I would like to open it up to questions now, and feel free to direct it to either or both of us.
Just
Yes.
a general question. One of you referred to just all the political discussion that took place in recent years. And of course, in the popular press, there's a lot about actual damage done to UCSF or delay of programs or loss of experts. And I was just wondering whether that was really true, those reports. You know, was UCSF damaged? Was there delay in your experiments and programs?
Well, I don't.
Oh, you can just
You wanna-
use the lectern microphone.
I think the answer is yes. I think every university in the U.S. was felt the impact of that. It did not affect my work directly, or I think Dr. Mancani's, but what basically happened is that a whole slew of scientists who worked on embryonic stem cells had to find a different way to get their research funded. And initially, this was an epicenter of that kind of research, and initially there was not light at the end of the tunnel. There wasn't a clear way to see how it was going to happen, and people left the university and went to other countries where such stem cell research was funded. It's since been rectified to some extent. You may have heard of CIRM, the California Institute of Regeneration Medicine, and that is a state-run institution that has decided to fund stem cell research. And so although the federal government stopped, CIRM arose, which is a huge boon to stem cell and regenerative medicine research in California. So that's a positive. The new administration in Washington, the Obama administration, has made strides to get embryonic stem cell research, as well as other kinds of stem cell research, supported. It's been difficult, and even today, it's a one step forward, two steps back kind of situation. But I think now people are feeling the effect still, but people are staying, and UCSF is extremely strong in stem cell research again, and it's partly because many people stayed, many new people came, and CIRM has helped a lot to fund the research while the federal government is deciding what it's going to do. Yeah.
Could you talk a little bit about Zometa? What is that really?
About what? Sorry?
IV Zometa?
Well, I mean, I think there's a lot of research using bisphosphonates to alleviate osteoporosis. And there has been a little bit of research trying to determine whether or not that has an effect on the cells that form bone, or whether it's really affecting the cells that resorb the bone. Is it working on the osteocytes or on the osteoclasts? And actually, honestly, even though there's a lot of data on both sides of that question, I don't think it's been definitively determined. Is that what you were hoping for? Okay. Yes.
Is the insurance industry helpful in terms of approving any of these procedures with patients?
Or funding it?
Most of what we talked about is experimental, and it's still ways away from being an established clinical approach that means that it's in the insurance books, and they'll look it up and see that it's called Procedure X and cover it. So I would say not yet. I would say most of this work that we've discussed is in a stage where it's experimental and part of clinical trials maybe, or part of laboratory work, but not yet covered by insurance. We expect that it will be, though, because part of what we envision is that these approaches will be economically superior to some of the approaches that exist. So a lot of money and time goes in on one end to develop these projects, but we hope that they'll prevent problems and make for more efficient ways to solve them later. So we anticipate that insurance companies might actually like it.
During the process of actually doing the clinical trials to validate these technologies, typically insurance won't cover. Those trials are typically funded by the companies that are producing the product or the universities that are sponsoring the research.
Oh, the question was in limb regeneration, for example, in newts. Okay, so the cells proliferate and grow, but how do they know what shape to make? And that's a huge area of research, and the bottom line is we understand very little. The cells don't, it's not that they only know what shape to make, but they actually know exactly where they are along the limb. They know how thick it's supposed to be and taper, and when do you decide to start making wrist instead of forearm bone, and so forth. And it's one of the great questions really in developmental biology and in regeneration biology. And you can see that for all the aspects of this that we talk about, there are huge questions that are still open. And one approach, the approach that I talked about, relies on the fact that maybe it's a default mechanism, and that if we can just unlock the some of the roadblocks that exist in our bodies, the cells will know what to do, and we won't have to figure it out because they clearly know what to do in those animals.
Dr. Makani, you mentioned Cytograph, that company. Are they still in the developing stage, experimental on creating those blood vessels for protein, or are they using it now?
So, well, the most recent information I had was that they were in clinical trials, and that the trials were based in Europe and perhaps in South America at that time. And I don't know whether they have gotten FDA approval for the product here in the U.S. yet.
How about skin? Are they using? I know they're using it some for burn victims, but I mean, is that being used right now?
Yeah, there's actually there's actually probably more research that's been done on skin substitutes and skin regeneration than there is on bone, and I confess to not being really current on that area, but I'll tell you what little I know, and I'm sure that Dr. Pomerantz can build on that. There are companies that are producing constructs in culture where they're producing sheets of cells. And you can get those delivered. They can either be cells that are from some universal donor, or they can be patient's own cells developed in tissue culture and then sent back after a number of weeks. So there are some skin substitutes that are currently available. They have some drawbacks associated with them, so they're not perfect. And they also are extremely expensive. But they're a first step towards a skin substitute.
Do you see yourself doing anything as far as researching that, or just the focus is stem cell?
I don't know if anyone's doing keratinocyte research here.
I'm sure, but I'm not sure who is. And I think the current state of the clinical applications really get back to one of the things Dr. Mancani was talking about. Skin is not just a layer of cells. Skin is a complex organ with multiple different cell types. It's thick. It has a epidermis that has cells at different stages. It has a dermis. It has blood vessels. It has cells with different functions. And so the sheets of cells that we currently have can cover a wound and prevent fluid loss and maybe protect from infection to some degree, but they're not the durable tissue that skin is. And I think, as far as I know, that is still very much in the research development stage. And we're waiting for the day when it exists, but not yet.
When you do this bone reconstruction, you are saying inject the bone cells and then let it grow there. How do you stop them from being cancerous later?
That's a great question. So, one approach has been to, from the standpoint of using adult-derived cells, is really to try to grow them in conditions which are not going to direct them towards becoming tumors. That's one approach. But there's actually no really good way to confirm that or not. We have an extensive experience in our lab and with my other group at the NIH, where we've done thousands of experiments and have never seen evidence of a cancer. So, our thought is that that's probably going to mean that it's not going to be a problem in a patient that gets it. But it's hard to prove a negative. So really all we have to go on is our experience at this point.
In regards to osteoporosis we were talking about, could injecting be used as preventive measures? Or
Right, that would be the ideal. Yeah, I mean, if you could identify a patient that was at a high risk of osteoporosis or was showing early signs of it, and you had a way to deliver cells to them that would help the bone regenerate, that would be an ideal preventative measure.
Do these have any application in arthritic joints or beginning of arthritic joints?
Yeah, I think so. I think very much so. In fact, there's been a lot of interest in trying to replace the denuded cartilage in arthritic joints by groups around the country, and there's been some success with that. So that's really an area of active research, whether it's using cells that are already committed to being cartilage or whether it's using stem cells which are not yet committed to being cartilage. A lot of groups have an interest in that. And actually part of the interest is economically driven. I mean, it's an area that has a lot of insurance reimbursement associated with it. So those groups that are involved in that have a lot of sort of economic inducement to be involved in the research and to try to push it forward. Well, thank you very much for coming out.
Thanks.